without evidence of infection or autoimmunity, and certainly there are no high-titer autoantibodies or antigen-specific T cells in these disorders. As you'll see, if we can have the next bullet, there's dramatic evidence of inflammation in these disorders. So here are some examples of, in the periodic fever syndrome, some of the images that perhaps illustrate the dramatic inflammation that some of these patients have. So shown here on the left is a laparoscopic view of the peritoneal cavity of a 7-year-old girl from California whom we saw a few years ago who has TRAPS, the TNF receptor-associated periodic syndrome. And this is one of the periodic fever syndromes that we'll be talking about today. She had had repeated episodes of peritonitis over the course of her young life and actually had developed adhesions in the peritoneal cavity, which are shown here, because of this repeated inflammation, this repeated sterile peritonitis that she had. Second image here is actually an echocardiogram taken of one of our patients with familial Mediterranean fever, a young man from Baghdad, Iraq, who was sent to us via London and then the Mayo Clinic with diffuse amyloid deposition due to FMF, and we'll talk a little bit about that. It's rare to see amyloid deposit in the heart in FMF, but in this particular case it did, and in fact this young man was in heart failure at the time we saw him with an ejection fraction of around 37% at the age of, I think he was 18 at that time. And here's an image of a patient with NOMID, the neonatal onset multisystem inflammatory disease, another of the recurrent fever syndromes that we'll be talking about. And this is basically a magnetic resonance image taken of the brain of one such patient. And you can see all the whitish areas here represent inflammation. This is chronic aseptic meningitis in this child. It leads to mental retardation, blindness, and deafness in these patients. So it's really a very serious and debilitating problem for patients who have this condition. In any case, as Francis mentioned, really we saw this as an opportunity, having diseases that are caused by single gene disorders, to identify what those genes are, to learn about new genes and pathways that regulate inflammation in humans. Because after all, if these kinds of clinical conditions result from those genetic mutations, they must have some important role in human biology. And so we thought that that was a critical insight. And these diseases also serve as the prototype for a group of diseases that we now call the systemic autoinflammatory diseases. These are a group of diseases that span a greater spectrum than just the hereditary fever syndromes, but they have in common the fact that there is unexplained inflammation, either recurrent or chronic inflammation, in the absence of high-titer autoantibodies or antigen-specific T cells. And it turns out that a lot of these disorders are in fact disorders of the innate immune system as opposed to the adaptive immune system, which has abnormalities in the more traditionally recognized autoimmune diseases such as systemic lupus or rheumatoid arthritis. So, in any case, the first patient that actually got me interested in these diseases was a man that I saw a number of years ago. It was actually 24 years ago at this point when I was a beginning fellow here at the NIH back in 1985. And he was, at that time, in his early 20s, named Sarkis, and he's of Armenian ancestry. And he presented to us, basically, with a history of episodic attacks of mono or quasi-articular arthritis, the knee, or the ankle, dating back to infancy. And he had had these attacks roughly once a month for his whole life. So he had had, at the time that I saw him, hundreds of attacks. During these attacks, he would have fever and an erythematous rash over the involved joints with massive joint effusions. And when he would have his joint aspirated, as sometimes happens if he happened to see a rheumatologist, there would be loads of polymorphin leukocytes in the synovial fluid. on the order of 100,000, which is what you would see in septic arthritis. The attacks would last on the order of several days. It would occur about once a month, and he had minimal response to corticosteroids. Now, at the time that I saw this man, I actually didn't know what he had. He was referred to us at the NIH New Patient Clinic as an unknown, and he was also an unknown to me. But fortunately, my attending was Paul Plotz, who suggested the diagnosis of FMF just based on the Armenian ancestry. And in fact, there was a fellow from Israel who happened to be in the lab that I was working in who said that this was obvious, that it had to be FMF, because they see it all the time in Israel. And it was a classic textbook case presentation. So this had sort of gotten me interested in FMF, and of course I learned as I started following Sarkis that in fact this is caused by a single autosomal recessive gene, and at least back at that time in the mid-1980s, there was a tremendous amount of excitement, at least in part of it incited by Francis with the work that he was doing on cystic fibrosis, that in fact we had the opportunity, using the new tools of human genetics and positional cloning, to identify some of the genes that underlie these conditions. And in that way, we could, without entertaining a lot of perhaps wild goose chase hypotheses, we could actually cut through the Gordian knot and be able to get to the heart of the matter and figure out what's going on in these patients. So that really appealed to me. And just for those of you who aren't familiar with FMF, and there could be any such people in this audience, just a few images that perhaps will illustrate the features of FMF. So just by a little way of background, the attacks of FMF usually last on the order of 1 to 2 months.